Hi, my name is Ian Dickey. I'm going to be talking about work that was done in my lab by Sarah Sapsford on the ecosystem impacts of coinvasive ectomycorrhizal fungal communities and plants. This is based on a general concept that was introduced by David Reed back in the 1990s that the world can be considered to be a mosaic of different mycorrhizal types. And although this idea is quite an old idea, it remains really relevant today. So, for example, you can see the recent paper by Steidinger, where mapping the world mycorrhizal dominance of ectomycorrhizal and arbuscular mycorrhizal vegetation was used to help understand ecosystem function. And so this has led to a general question of what are the differences between ectomycorrhizal and arbuscular mycorrhizal vegetation in terms of ecosystem function. And that's been approached through modeling efforts by Orwin and Phillips, through more empirical studies, things like Averell's study looking at carbon and nitrogen ratios around the world, and also through very detailed ecosystem measurements, particularly, for example, this work by Talbot looking at the abundance of soil enzymes involved in nutrient cycling and showing that they're correlated with ectomycorrhizal fungi. Or similarly, this work by Sturkenberg, where they trenched plots and showed that by excluding ectomycorrhizal fungi, they were able to change the soil enzyme profile. And then, of course, all this can also be approached through a genomic lens, looking at the genes involved in producing these enzymes and breaking down particular compounds. From all that work, we've had some emerging concepts. One is that interactions between mycorrhizal fungi and saprotrophs are really critical. Another is that there are specific enzymatic functions that are driven mainly by ectomycorrhizal fungi, but that these functions are contingent on the fungal community composition, what specific species of fungi are present. However, previous work has been somewhat limited in focusing on endpoints, that it's not generally looked at how these processes develop when an ecosystem changes between mycorrhizal types. And so that's been a real focus of my group recently, is looking at ectomycorrhizal invasions as a model for understanding what it means to be ectomycorrhizal as systems switch from being arbuscular mycorrhizal into ectomycorrhizal dominance. And the system we're using most widely is pine invasions. Pine invasions are a great system to work in. They're a huge problem in New Zealand, but they're also found throughout the world. This is a very similar invasion of Pinus contorta in Argentina and another example in South Africa. Now, I've been working on this for a while, and so about 10 years ago, we showed that the invasion of pine in New Zealand is driven mainly by fungal co-invasion, particularly some expected species such as Amanita and Suillus, but also this interesting one. The dominant fungus we found back in the 2010 study was the Canthorelales species, which we've now recently identified thanks to a collection by Tom Bruns as actually being a Cystotrema in the ectomycorrhizal section of that polyphyletic genus. So today's talk, what I want to do is update on that work, looking at the succession of fungi, not just what it's in a mature pine stand, but how do they develop over time, incorporate the saprotrophic community into our understanding of this process, and then look at pine removals where we've removed this invasive species and then seen how the ecosystem recovers or doesn't recover, which is a very management-oriented question, but has great implications for understanding the role of ectomycorrhizal fungi in ecosystem function. So the first study was published earlier this year. This was a study of a pine density gradient, so looking at succession of fungi as the pines filled in a landscape. We had 13 plots along a density gradient of pines. We actually took tons of samples. So we took 24 samples per plot so that we could look at diversity of ectomycorrhizal and saprotrophic fungi using next generation sequencing and do it at multiple different spatial scales. To summarize the results really quickly, this is an ordination of the ectomycorrhizal community. And what Sarah's done here is shown the uninvaded grasslands in light blue shading to darker blue as pine density increased. And you can see there's a shift as you go across an ordination from no pine to heavy pine. You also see that the size of the ellipse, so each ellipse represents all the points within a plot, and you can see those ellipses become smaller, suggesting the fungal community is becoming much more homogeneous as pines invade. And that's true both for ectomycorrhizal fungi here and on the other side for saprotrophs, showing a very similar pattern. In terms of the fungal community, it's an interesting succession. What we're seeing is, as expected, Suillus is really important in early succession. So this is looking at basal area of pine and the invasion of different fungi. What we see is Suillus comes in first. It's the only fungus that's consistently present in grasslands, but it's pretty rapidly followed by Trichiloma and Cystotrema, and Cystotrema becomes the dominant fungus in the ecosystem. Suillus remains present, but is never very dominant. And then lastly, the ecosystem develops more fully, and you get ammonite and other fungi coming in into quite a diverse community of ectomycorrhizal fungi. But overall, the main pattern is an enormous loss of fungal diversity. What we're seeing is at the point level, a decline from around 110 down to about 50 species of fungi. And in the plot level, or gamma diversity, going from around 650 down to around 300 species of fungi. So it's more than a 50% loss of fungal diversity. That's a little surprising. What's going on here? If ectomycorrhizal diversity is going up, how is total fungal diversity going down? And it's just a numeric thing. There is an increase in ectomycorrhizal diversity, but it's numerically very small. You go from around 3 or 4 species up to around 8 species, whereas the loss of saprotrophs and other fungi is huge. We also see a shift in the dominance of fungi in the system. This is looking at the basal area of pine and the proportion of DNA reads into different functional groups. And what you see is this green bar shows the increasing ectomycorrhizal dominance from being basically absent to making up more than 60% of the soil fungal community, while saprotrophs and undefined fungi, those with multiple functions, decline pretty rapidly. There are, however, some saprotrophs that increase. This is a heat map of different saprotrophic taxa. And you can see that there are some, as you look across increasing basal area, that actually go up in abundance, shown as the deeper purple colors, particularly this thing Saginomella. Turns out to be a real dominant fungus once pines invade. So that suggests that pine is really having profound effects on the saprotrophic community and potentially nutrient cycling. But to understand that, we decided to move into a system looking at legacies. And the nice thing with legacies is that managers have been removing pine, and that gives us the opportunity to see how ecosystems recover, and in doing so, link changes in the fungal community directly to ecosystem function. So what Sarah has done is to establish plots across gradients of density at removal, from very open to very dense stands, and across gradients of time since removal, from very recently controlled to controlled up to about 13 years ago. She's taken the approach of trying to understand the effect of those two drivers, time since removal and density at removal, through biotic and abiotic pools of fungi, bacteria, and soils, and looking at the impacts on the soil enzyme profiles. So what we did was we did an ordination on each one of those groups, and then we retained those axes that explained more than 8% of the variance. So we took the approach of doing ordinations on each one of those potential drivers. This is looking at an ordination of the fungal community. And what I've done is I've plotted all the plots. The size of the point indicates the density. So the larger plots had more pine. And the color, shading from dark green to sort of tan color, indicate how long ago it was controlled, with the darkest green being the most recent controls. What we saw was that the fungal community could be described by 3 axes, but it was really dominated by Cystotrema, and Saginomella, that saprotroph that responds positively to pine, and a few other fungi. We found that fungal community was very strongly driven by time since removal, but not by density at removal. In terms of what species are actually present, what we see is in the ectomycorrhizal community, when it's a very recently controlled stand, it's a diverse community of fungi. As time goes on, most of those fungi drop out. Interestingly, Suillus, which was the first fungus to come in, is also the last to be lost. In fact, we see it out to at least 12 years showing up in the soil DNA. Saprotroph communities are also recovering. There's the Saginomella, and that was dominant in pine invasions earlier, and again, it's dominant in this study, and then it's dropping out of the system, while a number of other saprotrophic species are increasing. When we looked at bacterial communities, we also found that they were driven by time since removal. It took a number of axes to describe them, and even with 4 axes, we're only capturing about 62% of the variance, because bacterial communities are quite variable in the system, but there is definitely an effect of the ectomycorrhizal invasion. Lastly, we looked at soils, and again, this came out somewhat complicated, but it falls down to three axes. We found that soil was described by one axis with basically nitrogen and organic matter, a second axis driven by pH and base saturation, and then a third axis, and this is important, driven by phosphorus availability and the carbon to nitrogen ratio. Of those three axes, only phosphorus and the carbon to nitrogen ratio, that is axis 3, was really driven by any pine factor, and it was driven by density at removal, not time, suggesting it's not recovering since the pines have been removed. Now, what about soil enzymes? Again, we did an ordination to simplify the data. What we found was that soil enzymes could be described by three axes. One that was really dealing with enzymes involved in cellulose and chitin degradation, another described by lactase and acid phosphatase, and then the last axis driven by manganese peroxidase. To link these together, we did a model simplification. I'm just going to show graphically what came out as significant drivers. The first enzyme, this is the cellulose and chitin degradation enzymes, was driven by a pretty complex combination of factors. The fungal community, bacteria, and soil factors were all important, but it was positively driven by specific groups of bacteria, the second axis of the ordination, low pH and high organic matter, and the effect of ectomycorrhizal fungi was largely negative. Axis 2 of the ordination, which was lactase and acid phosphatase, was also driven by bacteria, but by other groups of bacteria and soil organic matter. The effect of ectomycorrhizae overall was that as ectomycorrhizae increased, we saw a decrease in this axis. But interestingly, it was positively correlated with the fungal community axis dominated by Cystotrema and Wilcoxina, suggesting that there may be species-specific effects on lactase and acid phosphatase. But overall, we see that it does increase with time since removal, showing a recovery over time. Lastly, we looked at the third axis of the ordination, which was the manganese peroxidase enzymes. Here we found that there was a strong effect of density at removal and a negative effect of time since removal, showing it is recovering over time. This is consistent with high organic nutrient cycling under dense pine. However, we are not seeing a direct link to ectomycorrhizal fungi. In fact, what we're finding is that the axis associated with Cystotrema and Wilcoxina is negatively correlated with this aspect of enzyme function. That's quite confusing. We expected this enzyme to be very much linked to ectomycorrhizal fungi, and it may be linked, but linked to difficult to detect patterns. What I suspect might be occurring is that low abundance fungal taxa, which are not playing a major role in the fungal ordination, may actually be really important in driving this enzyme. That's going to be a real challenge for us to pick out in future research, trying to understand what is driving manganese peroxidase in these systems if it's not any of the dominant fungal players. But overall, what the results show is that ectomycorrhizal effects on function are complex. They involve strong suppression of saprotroph communities and indirect and direct effects via soil nutrients, bacteria, and saprotrophic fungi. A number of the effects are species-specific. For example, we saw that opposite effect of ectomycorrhizal dominance and the axis describing Cystotrema abundance, so that there are important species-specific effects on ecosystem function. From a management view, our results do have some good implications. They suggest that soil biological function is slowly returning. However, there are changes in soil chemistry, particularly, that are persistent and driven by density, not showing any sign of recovery. So that's a bit of a challenge for the future. On that note, I am in the process of recruiting a postdoc and a student. So if you are interested in this area of research, please do let me know. And lastly, I'd just like to reiterate that both these studies were led by Sarah Sapsford. She's just in the middle of moving to an early career research fellowship at Murdoch University in Perth, which is really great to see. So hopefully we'll be seeing more from her in the near future. And acknowledge my funding from the Ministry of Business, Innovation, and Employment's Winning Against Wildings program. Thank you for your time.